Imagine designing the tiny machines inside living things, proteins, almost as easily as typing an email. Now imagine making that power grow exponentially. Not just making things a little better, but unlocking entirely new capabilities, kind of like how computer chip power just exploded over the decades.
Right. Moore's Law, but for biology, maybe.
Exactly. Well, this deep dive is about how these massive AI models, the kind that learn human language from reading, like the whole internet.
Yeah, the LOM.
Are now learning the fundamental language of life itself, the actual sequences of proteins.
And the crucial part, the really exciting bit, is how applying this principle of scaling to these biological models could let us design and build biological breakthroughs way faster than nature ever could.
So welcome to the deep dive. We've got a stack of material here looking at a potentially massive development where AI meets biology.
That's right. And it really centers on this idea of scaling laws. The principle that if you make AI models much bigger, feed them way more data, use more computing power, you get predictable improvements, often with these surprising new abilities popping up. And the hypothesis is, does this apply to proteins too? Not just text or images.
And that's a huge deal because designing proteins from scratch has always been incredibly hard.
Notoriously difficult, yeah.
So our mission today is to unpack what these scaling laws actually are, understand why protein design has been such a tough nut to crack, and then really dive into a breakthrough from this company, Profluent Bio, with their model called ProGen3. It seems to offer some pretty compelling evidence that, these scaling laws are working in biology.
Think of it as getting a handle on a moment that could genuinely shift the whole landscape of biotech.
Yeah, sort of a shortcut to understanding something potentially game-changing.
We're basically asking if the engine that drove progress in large language models can now, power a similar leap forward in engineering molecules.
Yeah. Okay, let's really dig into this scaling laws idea first, because it feels fundamental to the whole story here. You've probably heard about or maybe even used large language models like the GPT series or Google's PaLM.
Sure, they're everywhere now.
The reason they got so incredibly good so fast in the last few years wasn't just about, clever new algorithms, although that helped.
Right.
A massive part of it was just making them, huge, gigantic models.
Exactly. That observation, which people started calling scaling laws, is basically this.
Yeah.
If you plot how well a neural network performs, specifically its error rate or loss against things like its size, meaning the number of parameters, the amount of data it's trained on, and the compute power used, you see this really consistent, predictable trend. It follows a power law.
A power law.
Yeah, meaning as you scale up those inputs, size, data, compute dies, the error rate drops smoothly, predictably. It's not just a little bit better each time. It's this strong, almost physics-like relationship, at least in certain setups.
And this wasn't just a theory, right? People actually showed this works.
Oh yeah, researchers demonstrated this rigorously across many orders of magnitude for language models, especially around 2020. It basically gave AI researchers a clear recipe. Need better performance, scale up. Model, data, compute. Keep pushing the scale.
But you said it gets even more interesting than just, lower error rates.
Right, and this is maybe the most exciting part about scaling. It's not just about making the AI better at what it already does. Once you pass a certain size threshold, these models start showing emergent capabilities. Entirely new skills or behaviors that nobody explicitly programmed in. They just arise, seemingly spontaneously, just from the model seeing enough data and building this deep general understanding.
Can you give an example from the language models? Like what kind of new skills?
Sure. Think about it. A smaller language model might be okay at finishing your sentence. But then you scale it way up, going from maybe millions of parameters to billions, like GPT-2 versus GPT-3, or even hundreds of billions like PaLM. And suddenly, these huge models can do things like few-shot learning. You give them just a couple of examples of a new task, like translation or summarizing, and they can just do it, even though they weren't specifically trained for that task before.
Wow.
Or they show complex logical reasoning, or they can explain abstract concepts, even understand and explain jokes like PaLM did. These abilities weren't taught. They emerged from the scale.
So scaling isn't just turning the volume knob up. It's unlocking completely new features on the stereo.
That's a great analogy.
Yeah.
It fundamentally changes what the AI can do. It provided this powerful roadmap for AI research. If you want to push the boundaries, focus on achieving scale.
And doing it efficiently, right?
Right.
You mentioned compute optimal training.
Exactly. You don't just want a huge model if you don't have enough data, or tons of data with a tiny model. The scaling laws helped figure out the sweet spot, how to balance model size and data size to get the most improvement for the amount of computation you can afford, like DeepMind's chinchilla model showed for language.
And are there like technical tricks that make these huge models feasible to train? The material mentioned something about mixture of experts.
Yeah, mixture of experts, or MOE, that's a key architectural innovation. So a standard big model, a dense model, uses all its billions of parameters for every single input it processes, which gets computationally very expensive.
Whoa.
And MOE model is different. It has different groups of parameters called experts and a little routing system. For any given input, it only activates a fraction of those experts.
I see.
So you can have a model with a truly massive total number of parameters, hundreds of billions, even trillions, but the actual computation needed for each step is much lower, closer to that of a smaller, dense model. It lets you scale up the parameter count much more efficiently.
Got it. So the big takeaway for AI was scaling works, it's predictable, and it unlocks surprising new abilities.
Exactly. A powerful formula.
Okay, now let's completely switch gears. Biology. Because the whole point here is applying these AI scaling ideas to the wheel of molecules, specifically proteins.
Right. The essential workhorses of life.
They really are incredible, aren't they? These tiny machines in every cell.
Absolutely. They're the enzymes that make almost every chemical reaction happen. Digestion, building DNA, you name it. They're antibodies fighting off disease. They build our tissues, like collagen. They transport stuff, receive signals. Just incredibly versatile.
And they're built from a surprisingly simple alphabet, right?
Yeah, just 20 different building blocks, the amino acids. They link up in a chain, like letters in a word. But here's the critical part. The specific sequence of those amino acids dictates how that chain folds up into a unique, complex 3D shape.
Okay.
And it's that precise 3D shape, its bumps, grooves, surfaces, that determines what the protein actually does, whether it can grab onto another molecule or speed up a reaction or slot into a structure.
So if you want to design a new protein to do a specific job, you need to figure out the exact amino acid sequence that will fold into the right shape and have that desired function. Why has that been so incredibly hard?
Well, this is where you run into the just insane numbers. The main problem is the sheer size of what's called the sequence space, basically. All the possible protein sequences you could imagine making.
Okay.
Even for a pretty short protein, let's say just 100 amino acids long. You have 20 choices for the first position, 20 for the second, and so on. So the total number of possibilities is 20 times 20 times 20, 100 times. That's 20 to the power of 100.
And how big is that number, really?
It's mind-bogglingly huge. It's vastly larger than the estimated number of atoms in the entire observable universe.
Wow. Okay. That puts it in perspective. It's not just big, it's fundamentally unimaginable.
Exactly. So trying to find a useful protein by just randomly guessing sequences, it's completely hopeless.
Yeah.
Utterly impossible. Most random sequences would just be floppy, useless strings. They wouldn't fold properly, let alone do anything useful.
Like hitting random keys on a keyboard and hoping to rate Shakespeare.
Precisely. The odds are effectively zero.
And that's why traditional protein engineering has been such a slow, painstaking process.
Absolutely. The main approaches have been things like directed evolution, where you take a protein that already exists, make lots of tiny random changes, and then test millions of versions in the lab to find slightly better ones. Mimicking evolution, but accelerated.
Right.
Or rational design, where scientists use their knowledge of protein structure to make educated guesses about changes, then test them. Both are really slow, really expensive, need tons of lab work, and usually only explore tiny variations around proteins we already know, designing something totally new from scratch. That's been the grand challenge.
So there's this incredible potential for custom molecular machines, but we've been stuck because the search space is too big and the old methods are too slow.
Exactly. Bottlenecked by complexity and experimental limits.
And that's the problem that AI, especially AI using these scaling laws, is now trying to tackle for proteins.
That's the promise.
Okay, so this brings us directly to Profluent Bio and their work with this ProGen3 model. They basically looked at the success of scaling in language AI and asked,
What if? What if those same scaling principles work for the language of proteins? Could making a protein-generating AI model dramatically bigger unlock similar leaps in its ability to design actual functional biological molecules?
That was the core hypothesis they tested.
Yes. So Profluent built ProGen3, which isn't just one model, but a family of generative models specifically designed to create new protein sequences. And they scaled it up massively. The biggest one they reported in this work has a huge 46 billion parameters.
46 billion. How does that stack up against other protein AI models?
It's the largest protein-generating model reported so far, by quite a margin. Many other state-of-the-art models, whether for predicting structure or generating sequences, have been more in the range of hundreds of millions, maybe a few billion parameters.
Okay.
Hitting 46 billion is a really significant jump, a direct application of that go big philosophy from language AI.
And scaling the model means you also need to scale the data, right? You can't train a giant model on a small data set.
Absolutely critical. Otherwise, the model just memorizes the data it saw. ProGen3 was trained on a correspondingly massive dataset, over 3.4 billion protein sequences,
Wow.
which translates to about 1.1 trillion amino acid tokens, the individual building blocks. They curated this dataset, calling it the Profluent Protein Atlas V1, pulling together sequences representing the known diversity of life's proteins.
And how does that data size compare?
It's significantly larger than what previous big protein language models used.
Yeah.
For instance, Meta's ESM2 used data around 15 billion tokens, I think. Salesforce's earlier ProGen2 used less than ProGen3 as well. So it's a big step up in data scale, too.
And you mentioned they curated it carefully, not just quantity, but quality and diversity.
Yes, that's vital for training a good foundation model. It filtered out low-quality sequences, reduced redundancy, and crucially, they balanced the dataset. Natural protein databases are heavily skewed towards common organisms like E. coli or humans.
Right.
By balancing, they made sure the model saw a good representation of proteins from across the tree of life, including rarer protein families. This helps the model learn more general rules about proteins rather than just getting really good at mimicking the most common types.
And they use that compute optimal approach, balancing model size and data.
Yes, they followed those principles, learning from work like DeepMind's Chinchilla in the language space to get the most out of their computational budget. And they also use that sparse mixture of experts architecture we talked about earlier.
The MOE trick to handle the huge parameter count efficiently.
Exactly. It lets them have that headline 46 billion parameter number while keeping the actual cost of training and using the model manageable. But here's the absolute key takeaway from the Profluent work, the real breakthrough.
Okay.
They didn't just stop at showing that the bigger models got better scores on computational benchmarks, like lower loss. They actually took the protein sequences generated by the AI, synthesized them, and tested them in a real wet lab.
They actually built the proteins.
Yes. This is the first really large-scale experimental proof that scaling up protein generation models makes them genuinely better at creating proteins that actually work biologically. That's the crucial link that was missing.
Okay, so the proof is in the pudding, or in this case, the protein. What did those wet lab experiments actually show? What were the benefits of scaling ProGen3?
Well, they saw really clear, measurable improvements directly tied to the model size, mirroring what happened with language models, but now reflected in biological function.
Like what specific improvements?
One really striking finding was about accessing new protein families. The largest model, the 46 billion parameter one, could successfully generate viable protein sequences from families that the smaller models, even multi-billion parameter ones, completely failed on. They just generated junk for those rarer families.
So the big model could reach parts of the protein universe that smaller ones couldn't.
Exactly. It suggests the 46B model learned enough general principles from that huge, diverse data set to sort of extrapolate, to explore more distant corners of the sequence space. Smaller models might just remix common themes they saw a lot, but the biggest one showed signs of a deeper, more general understanding, allowing more creative leaps.
So more novelty, reaching into new design spaces. What else?
Another major benefit was increased diversity in the designs. Profluent measured this by generating batches of sequences from each model size and seeing how many distinct types or clusters they got. The ProGen3 46B model produced 59% more unique sequences, defined as sequences differing by at least 30% compared to their 3 billion parameter model.
Almost 60% more diversity just from scaling.
Yeah, and compared to an even smaller 339 million parameter model, the diversity jump was nearly 200%.
Why is diversity so important in protein design?
It's absolutely crucial. When you're trying to find a protein with very specific properties, maybe an enzyme that works perfectly at 70 degrees Celsius, or an antibody that binds incredibly tightly to a virus, you need to explore a wide range of possibilities. If your AI just gives you slightly different variations on the same basic design, your chances of hitting that perfect optimum are much lower. More diversity means the AI is casting a much wider net, exploring a richer landscape of potential solutions. It dramatically increases the odds of finding truly novel candidates with exactly the properties you need, maybe even solutions a human designer would never have thought of.
Okay, so bigger models give more diverse, more novel sequences, even exploring new families. But the acid test, do these AI sequences actually work? Can they be made into real proteins in a cell?
That's the make or break question, right?
Mm-hmm.
Generating text is one thing. Generating functional biology is another level. And Profluent did extensive wet lab testing on this. They synthesized and tried to express many generated proteins from the different model sizes. And they found that a high fraction of the proteins designed by the larger models could be successfully expressed in standard lab systems, like E. coli bacteria. And importantly, they were expressed at levels comparable to natural proteins. This even held true for sequences that were very novel, sometimes only having 30% similarity to any known natural protein.
Wow, even the really weird ones worked.
Yeah, it means the AI isn't just spitting out random strings of amino acids. It has somehow learned, or internalized, the fundamental rules of protein folding and stability. It's generating sequences that are biologically plausible, sequences that cells can actually build in to functional proteins.
That's amazing. Like the AI learned the grammar and syntax of the protein language well enough to write valid functional code.
That's a great way to put it. But maybe the most compelling evidence for scaling laws applying to function came when they tested the AI's ability to design proteins for a specific purpose. They did this by fine-tuning the model.
Fine-tuning.
I
Like
know.
giving it some extra training on a specific task.
Exactly. They took small amounts of lab data, basically, lists of protein sequences, and their measured fitness scores for a particular property, like how stable the protein is or how active an enzyme is. They showed this data to the pre-trained models.
Okay.
The goal was twofold. First, could the model learn from this limited data to predict the fitness of new sequences? And second, could it actually generate new sequences that were even better than the ones it was trained on?
And how did scaling affect this?
This is where the adaptability of the larger models really shone. Even before any specific fine-tuning, the big 46B model was already somewhat decent at predicting protein fitness, just from the sequence it showed about a 33% correlation with the lab measurements.
Okay, not bad.
But after it was fine-tuned, or aligned, as Profluent calls it, with just a small amount of experimental data for a specific task, its ability to predict fitness for that task jumped dramatically to about 67% correlation.
Doubled its accuracy with just a little bit of real-world data.
Pretty much. And here's the kicker. The larger models got much better at this alignment process than the smaller ones. The 46B model wasn't just better initially, it was vastly more adaptable. It learned far more effectively from that small amount of lab data compared to its smaller siblings.
Ah, so bigger models are better learners when it comes to specific tasks.
Exactly. More sample efficient is the term. It strongly mirrors what happens with large language models. They become much better at few-shot learning and adapting to new tasks as they get bigger.
So the bigger the protein model, the better it can leverage its general knowledge to quickly master a specific design goal, like making a protein super stable using only a little bit of targeted lab data.
Precisely. It suggests the huge models have built up this really rich general understanding of protein biophysics. When you give them a little hint about what good looks like for a specific property, say, working in high heat, they can connect that goal to their vast knowledge base and figure out the sequence features needed, then generate novel candidates optimized for it. Smaller models just don't have that underlying depth to work with, so they don't improve nearly as much with the same amount of fine-tuning data.
That synergy between scale and adaptability seems incredibly powerful for practical protein engineering.
It really is. Profluent summed it up by saying the larger models more faithfully represent the principles underlying life's diversity and are more amenable to fine-tuning for functional goals. It's strong evidence that biology follows its own scaling laws, and bigger AI models lead to experimentally validated, better, more adaptable biological designs.
Okay, so designing proteins on a computer is cool, but as you said, the real proof is in the wet lab. Profluent really emphasized that they validated ProGen3's designs experimentally, showing they actually work.
Yeah, that's the crucial step, the one that bridges the gap between computation and biology.
Yeah.
It's non-negotiable.
Can you walk us through that workflow? How did they go from an AI sequence to a tested protein?
Sure. So the AI generates an amino acid sequence. Then you essentially print the corresponding DNA gene, usually by ordering it from a commercial DNA synthesis company.
Right.
Then you take that synthetic DNA and insert it into cells, often standard lab workhorses like E. coli bacteria or maybe mammalian cells, depending on the protein. You coax the cells to read that DNA blueprint and actually build the protein.
Okay, protein expression.
Exactly. Then you purify the protein and run experiments to see if it folds correctly and does what it was designed to do. Does it bind to the target? Does it catalyze the reaction? How stable is it? Profluent has its own labs set up to do this whole cycle, which is important for moving quickly between design and testing.
And they didn't just test random proteins, they focused on designing things with real-world applications.
Yes, they showcased results in two really impactful areas. First, designing brand new antibodies to therapeutic targets. They called this initiative Open Antibodies. They used ProGen3 to design antibodies aimed at proteins that are already targets for major FDA-approved drugs, like for cancers such as multiple myeloma.
Targeting something like CD38, which existing drugs hit.
Exactly that one. And the results were pretty stunning. A good number of the antibodies designed entirely by ProGen3 bound to their targets just as tightly as the existing clinically successful drugs.
Wow, as good as approved medicines straight out of the AI.
Yeah, but it gets better. Some of these AI-designed antibodies actually showed improved properties like stability or how easy they are to manufacture compared to the existing drugs. That's a big deal because usually in antibody development, there's this annoying trade-off. Making it bind tighter often makes it less stable or harder to produce.
Right.
But the AI seemed to find solutions that balance both binding and these developability properties really well. And importantly, it did this in a single design round.
Single-shot designs, meaning no lengthy optimization needed.
Exactly. Traditional antibody discovery involves years of screening millions of candidates, then painstakingly optimizing the best ones through iteration after iteration in the lab. ProGen3 generated competitive candidates in basically one computational step.
And were these just slight variations of known antibodies?
No, that's another key point. They were genuinely novel. When they compared the sequences, the AI antibodies typically only had around 80% similarity to any known antibody for that same target. And the differences were often in the most critical parts, the loops called CDRs, that actually contact the target.
So the AI found completely new ways to bind the target.
It looks like it. It wasn't just modifying existing keys, it was inventing entirely new keys that fit the same lock. From an intellectual property perspective, that's obviously huge too. These are potentially novel, patentable molecules. It really shows the AI's creative power in exploring that vast sequence space.
That's incredible for drug discovery. What about the other example you mentioned, gene editors?
All right, this is another area where protein properties, specifically size, create a real engineering bottleneck. CRISPR-Cas9 has revolutionized gene editing. We all know that. But the natural Cas9 protein is pretty big, about 1368 amino acids long. This size makes it tricky to deliver into cells for gene therapy. The preferred delivery trucks, these little viruses called AAVs, have very limited cargo space. Cas9 often barely fits, leaving no room for other necessary components.
So the challenge was to make a smaller, functional CRISPR.
Exactly. Profluent tasked ProGen3 with designing ultra-compact Cas9-like enzymes that could still cut DNA effectively.
And did it work?
It did. ProGen3 generated novel gene editing enzymes that were as small as 592 amino acids. That's less than half the size of the standard Cas9.
Less than half the size. And they actually functioned in human cells.
Yes. They demonstrated in the lab that these compact, AI-designed editors could successfully cut DNA targets inside human cells. This is potentially transformative for gene therapy because these smaller editors can easily fit into those standard AAV delivery vectors with room to spare. It overcomes a major hurdle.
It sounds like the AI isn't just mimicking nature, it's actually inventing new tools to solve specific engineering problems.
That's exactly it. Evolution didn't necessarily select for the smallest possible Cas9, but human engineers need one for gene therapy. The AI, trained on the principles of protein function but not constrained by specific evolutionary paths, could find these novel, compact solutions. Profluent had released an earlier version, OpenCRISPR-1, designed by AI, and these ProGen3 versions are even better.
So the wet lab results really back it up. AI scale leads to functional innovation designing novel antibodies competitive with drugs and ultra-compact gene editors that work. It's practical progress.
It really validates the power of this scaled, sequence-first, generative approach. Going from a design concept to a working protein candidate computationally, drastically shrinking the traditional design cycle.
Now, it's probably helpful to place ProGen3 in the context of other big AI tools in biology. It's not the only game in town, right? And they do different things.
That's a really important distinction to make, yes. ProGen3 fits squarely into the category of generative protein sequence models. Its main job is to create new, amino acid sequences to write new protein sentences, if you like. Earlier models in this space include things like ProGPT-2, Salesforce's ProGen series before this big one, and Meta's ESM2 language models. ProGen3 is essentially a scaled-up successor in this lineage.
Okay, so it generates sequences. How is that different from something like AlphaFold, which everyone heard about?
Totally different task. AlphaFold 2 and similar models like RosettaFold or ESMFold are structure prediction models. You feed them an amino acid sequence, usually of a protein that already exists or one you've designed, and they predict its 3D folded shape.
Ah, so they tell you what a sequence looks like.
Exactly. They were revolutionary because they suddenly let us see the likely structures of millions of proteins without needing slow, expensive lab experiments like X-ray crystallography. They answered the question, given this sequence, what's its structure? Hugely valuable for understanding biology, but they don't create the sequence itself.
Okay, so AlphaFold predicts the shape of a given sequence, while ProGen3 actually creates novel sequences in the first place.
Precisely. They're complementary tools. You might use ProGen3 to generate, say, 1 million potentially interesting new sequences for a target, then run the top candidates through AlphaFold to predict their structures and filter for ones that look promising or are likely to fold correctly before you go into the lab.
Got it. Are there other types of AI for protein design?
Yes. There's another important category, structure-based generation models. Things like RF diffusion work differently again. They might start with the desired 3D shape or protein backbone structure and then try to find an amino acid sequence that would likely fold up into that predefined shape.
So they design sequence based on a target shape.
Kind of, yeah. They're powerful for designing proteins with very specific geometric requirements, like fitting into a particular pocket or creating a specific binding surface. It's a different approach to the problem structure first versus sequence first.
And ProGen3 is firmly in that sequence first camp, just taken to a much larger scale.
Exactly. The significance of the ProGen3 work is showing that by massively scaling up a sequence-first generative model and validating it rigorously in the lab, you can achieve remarkable success in designing novel, functional proteins. While AlphaFold gives you the structure and RF diffusion builds from shape, ProGen3 demonstrates the power of learning the language of protein sequences directly at scale to write new biological blueprints that actually work.
Okay, let's zoom out. What does all this really mean? This confirmation that AI scaling laws work for proteins, ProGen3 designing functional molecules. How does this actually change things for biology, for medicine, for us? Why should people listening really care?
Well, fundamentally, it signals a shift. It's not just a slightly better tool. It changes the whole paradigm of how we approach biological engineering. We're moving away from just discovering what nature made towards intentional AI-driven design.
From discovery to design, as you said earlier.
Exactly. Think about it. Instead of sifting through nature, hoping to find an enzyme that does what you want, or spending years tweaking an existing one, you could potentially just specify your goal to the AI. I need an enzyme that breaks down plastic X at temperature Y and pH Z, or design me an antibody that binds super tightly to the specific part of a new virus. The AI then generates tailored candidate sequences. You're not just finding biology, you're authoring it.
And the implications for making new medicines seem enormous.
Absolutely critical.
Yeah.
Developing protein drugs, antibodies, therapeutic enzymes is currently incredibly slow, expensive, and has a high failure rate. Profluent showed they could design panels of novel antibody candidates computationally, basically overnight.
Right, the single-shot designs.
Yeah. Compare that to the traditional multi-year process involving animal immunization, massive screening, and laborious optimization. This could slash drug development timelines and costs. It might let us respond much faster to new diseases, tackle targets previously considered undruggable, maybe even enable personalized protein therapies designed for an individual's specific needs.
And outside of medicine, industrial applications.
Huge potential there, too. Enzymes are workhorses in so many industries: detergents, food processing, biofuels, textiles, chemical manufacturing. Companies always need better enzymes that can withstand specific industrial conditions. High heat, harsh chemicals, weird pH levels.
Things nature didn't necessarily optimize for.
Exactly. Finding or engineering those enzymes is tough. But a scaled AI like ProGen3 can be fine-tuned to design enzymes specifically optimized for those challenging industrial requirements.
So you could get AI to design the perfect enzyme for a greener laundry detergent that works in cold water, or one to efficiently break down waste plastic at a recycling plant.
Precisely. Or enzymes for capturing carbon dioxide, making agriculture more efficient, creating new sustainable biomaterials. Any area where a specific biological catalysis or function is needed. The AI can combine tricks learned from billions of years of evolution in entirely new ways to meet human-defined goals.
And these models themselves, they become repositories of biological knowledge.
That's a fascinating aspect. Just like researchers study large language models to figure out what they've learned about grammar or reasoning, scientists can probe these massive protein models. Their internal workings might reveal deep insights into the rules of protein folding, stability, and function relationships that are hard to see just by looking at natural proteins. They become these incredibly dense knowledge bases about protein science.
And could we see even more surprising abilities emerge as these models get even bigger?
That's the big hope. Follow the pattern from AI. As protein models scale further, maybe integrate more types of data structure, function, interaction data, they might spontaneously develop new capabilities. Maybe predicting how multiple proteins interact, designing whole protein complexes, perhaps even understanding or designing parts of cellular pathways. Developing a kind of intuitive physics for molecular biology.
It really sounds like we're heading towards true foundation models, but for biology.
That's the vision many are pursuing. ProGen3 is a major step in that direction. Imagine one giant pre-trained AI model that understands the fundamentals of protein sequence, structure, and function. You could then adapt it relatively easily for a huge range of specific biotech tasks, designing antibodies, enzymes, diagnostics, gene editors, without needing to train a new model from scratch each time. A unified AI engine for bioengineering.
Which can make powerful protein design tools accessible to more people.
Absolutely. It could democratize it. Instead of needing a PhD in protein engineering in a huge lab, maybe a biologist or chemist could use a powerful AI model through an interface to generate promising designs for their specific problem, much like people use AI image generators today. It lowers the barrier to entry and could spark innovation everywhere.
And you mentioned earlier the idea of coupling this with automated labs.
Yes, that creates a really powerful feedback loop. The AI designs millions of candidates. Robotic labs synthesize and test thousands of them automatically, and the results feed straight back into the AI to make it smarter. These robot scientist setups can explore the design space and optimize proteins at a speed and scale far beyond manual methods.
So AI becomes this central engine driving biological innovation, but this kind of power obviously raises some serious questions. Ethical concerns, safety, especially the dual-use problem.
Yes, this is where we absolutely need to be thoughtful and proactive. The same AI technology that holds immense promise for good curing disease, helping the environment, could potentially be misused to design harmful biological agents.
How could that happen? What's the specific risk?
Well, these large models are learning the deep rules connecting amino acid sequence to biological function. They understand what makes a protein stable, how it interacts, what gives it activity. A sufficiently powerful model could potentially use that knowledge to design a protein sequence that is, say, highly toxic to humans or crops, maybe easy to produce, and crucially, novel.
Novel meaning it doesn't exist in nature.
Exactly, and that's a key part of the concern. Current biosecurity measures, like the screening done by DNA synthesis companies, often rely on checking customer orders against databases of known dangerous pathogens or toxins.
Right, they look for known bad sequences.
But if an AI invents a completely new toxic protein sequence, one that's not in any database, it might slip right through those existing screens. An AI could theoretically design a protein that disrupts a vital biological process in a way nobody anticipated, or maybe makes an existing pathogen more dangerous.
So the very creativity of the AI becomes a potential security challenge.
It's a challenge that the AI safety and biosecurity communities are taking very seriously. There are ongoing discussions globally involving governments, researchers, and companies about how to mitigate these risks. Some ideas include enhancing DNA synthesis screening, maybe using AI itself to predict the potential function or risk of a requested sequence, not just checking against known lists.
What about controlling access to the most powerful models?
That's seen as a key near-term measure. Companies developing these cutting-edge models like Profluent are generally not releasing the full models openly right now. Instead, they're offering access through APIs or collaborations. This creates a checkpoint and makes it harder for someone with bad intentions to get their hands on the most capable tools. There's also talk about potential regulatory frameworks, maybe like export controls, and the idea of audit trails to track how these powerful biological AI tools are used, though that raises privacy issues too.
It sounds like a really tricky balancing act. You want to enable the good research, but prevent the bad.
It absolutely is. The goal is to find safeguards that are effective against misuse, but don't unnecessarily stifle the incredible potential benefits. And it's not just about malicious use. There's also biosafety, preventing accidental harm from an AI-designed protein that has unexpected side effects. Rigorous testing and risk assessment are crucial for any novel biological agent, regardless of how it was designed.
But AI could also be part of the solution here, right? Not just a potential problem.
Yes, definitely. That's the flip side. The same AI capabilities can be turned towards defense and safety. AI could potentially design antidotes or countermeasures to novel threats much faster than traditional methods. It could help scan vast amounts of biological data to detect suspicious sequences or emerging outbreaks. Experts are working on building safety and security principles directly into the AI development process, emphasizing responsible innovation and keeping humans in the loop.
Okay, beyond the safety aspects, it's clear this AI revolution in protein design is also stirring up a big commercial and strategic race.
Oh, absolutely. The ability to design novel, functional proteins efficiently is a massive potential market disruptor. The most obvious sector is pharma, and biotech protein drugs are already a huge market, hundreds of billions of dollars. Companies like Profluent are aiming squarely at designing the next generation of these therapies.
So AI-designed molecules could become the next blockbusters.
That's the bet. By speeding up discovery, creating genuinely new molecules that get around existing patents, maybe tackling diseases that were hard to drug before, AI could fundamentally change the economics and timelines of drug development, creating huge value through new intellectual property based on these AI-native proteins.
And this isn't just limited to medicine, is it?
Not at all. Think about industrial biotech companies using enzymes for manufacturing, food, biofuels, environmental cleanup. They all stand to gain hugely from custom-designed, more efficient enzymes. Agriculture could use AI-designed proteins for better crops or safer pesticides. Even material science could see new protein-based materials with unique properties. Anywhere molecular function matters.
So who are the key players jockeying for position in this new space?
It's a really dynamic mix. You've got specialized AI biotech startups, Profluent Bio, Evolutionary Scale, which came out of Meta AI, Generative Biomedicines, Absci, Cradle, Evozyne, all focused on building these advanced AI platforms for protein design.
Okay, the focused startups.
Then you have the big tech giants with deep AI expertise like Meta itself and Google Alphabet through DeepMind and Isomorphic Labs, who are also developing foundational protein models. And the cloud providers are crucial. NVIDIA with its BioNeMo platform, Microsoft Azure. They provide the massive computing power needed and are building specialized tools for bio-AI.
And what about the established pharmaceutical companies? Are they building their own AI platforms or partnering up?
It's a bit of both, and it's a big strategic dilemma for them. It's incredibly expensive and difficult to replicate the kind of massive AI infrastructure, hire the specialized talent, and tightly integrate it with the necessary high-throughput labs. So many big pharma companies are choosing to partner with or license technology from the AI biotech startups.
Like licensing access to ProGen3 or its designs from Profluent.
Exactly. Profluent is offering licenses and API access. This lets pharma tap into the cutting-edge AI capabilities without having to build it all from scratch. It's a classic build versus buy or license decision.
But the big ones must be trying to build some internal capacity, too.
Oh, for sure. The largest pharma companies are definitely investing heavily in internal AI teams. But the ground is shifting. The traditional competitive advantages, maybe having the biggest chemical libraries or the most extensive screening labs, might become less important compared to having the superior AI models and the most efficient pipeline for generating and validating novel AI-driven designs.
So the cost per successful discovery could drop dramatically, shifting where the value lies.
That's the potential disruption. The competitive edge increasingly comes from the power of the AI model, the quality and scale of the training data, especially proprietary lab data fed back into the models, and the strength of the intellectual property generated by the AI's novel designs.
It sounds like it really demands people who understand both sides, the AI and the biology.
Absolutely. The winning teams will be the ones who can fluidly bridge computation and experimentation. We're seeing the rise of these full-stack bioengineers, people who are deeply skilled in both advanced machine learning and molecular biology and protein engineering. That cross-disciplinary expertise is becoming golden.
So looking even further ahead now, where does this trajectory take us? What's the future of this AI-native biology?
Well, ProGen3 at 46 billion parameters is impressive, but it's almost certainly not the endpoint. If the scaling laws continue to hold, we should expect even larger models, maybe 100 billion parameters, maybe more in the next few years. And crucially, these models will likely be trained on richer, more diverse types of biological data, Not just protein sequences, but maybe integrating 3D structures, functional assay results, data about how proteins interact, where they are in the cell, maybe even genetic regulatory information. As the models get bigger and see more kinds of data, we should expect even more powerful emerging possibilities.
The outline mentioned designing entire biological systems, maybe even minimal cells. Is that science fiction or a plausible long-term goal?
It's definitely way out there, but it's a plausible extension of the core idea. If AI can master the language of single proteins, can it then learn the grammar of how proteins work together, how they're controlled by DNA, how they form pathways and networks?
Right, the system level.
Exactly. Design not just the individual parts, but how they interact dynamically to create a functional system. It's orders of magnitude more complex, would require even bigger models and more sophisticated training data, but the conceptual path is there. AI might eventually assist in, or even lead, the design of synthetic gene circuits, metabolic pathways, maybe even optimizing the components for simplified artificial cells.
Moving from designing a single molecule to designing a complex biological machine.
That's the grand long-term vision. Engineering biology at the systems level, maybe creating organisms optimized for producing valuable chemicals, cleaning up pollution, or acting as living sensors.
And that synergy with automated labs, the robot scientists, will just keep accelerating things.
Absolutely. That rapid design, build, test, learn cycle is key. AI proposes thousands of designs, robots build and test them at scale, data flows back to the AI. It's how you navigate that immense design space efficiently and find the truly novel solutions.
And could AI actually discover things that nature never did? Completely new biological principles or tools?
That's one of the most exciting possibilities. We saw ProGen3 design a compound CRISPR that's different from natural ones. As these AI models get even more powerful and explore the sequence function landscape more deeply, they might stumble upon entirely new ways for proteins to fold, or new catalytic mechanisms, or new ways to interact with cells that evolution just never happened upon or wasn't selected for. They could become engines of true biological invention.
It really does feel like we're entering an era of AI-native biology.
I think that term captures it well. An era where many new biological innovations, molecules, tools, maybe eventually systems are conceived and optimized computationally first, born from AI analysis and creativity, rather than solely discovered in nature or through slow lab tinkering. We're gaining the tools to become authors, not just readers, of the book of life.
What are the biggest hurdles still remaining?
Well, biology is still incredibly complex. Capturing all the nuances of how a protein behaves inside a living cell, dealing with interactions, toxicity, immune responses, dynamic changes, is a huge challenge for AI models. Making sure AI designs work reliably and safely in those complex, real-world biological contexts is paramount.
Right.
There's also the black box problem, or interpretability. Why did the AI make this specific design choice? Being able to understand the AI's reasoning could yield huge scientific insights, not just functional proteins. And, of course, the sheer cost of computation and the need for vast amounts of high-quality experimental data to train and validate these giant models remain significant bottlenecks. But the field is pushing hard on all these fronts.
Okay, so let's try to wrap this up. To synthesize everything we've discussed.
It seems clear that scaling laws, the principles that revolutionized AI for language, are indeed working for the language of life protein sequences. Models like ProGen3, trained on billions of evolutionary examples at unprecedented scale, can now design functional, novel proteins essentially from scratch.
Right, compressing evolutionary time into software cycles.
Exactly, offering potential solutions for medicine, industry, and basic science. And crucially, this isn't just theory. It's backed by large-scale experimental validation, showing that bigger AI models genuinely produce better, more adaptable biological designs. Biology has its scaling laws, too.
We really do seem to be at a remarkable inflection point. Information computation is starting to directly engineer physical matter at the molecular level, giving us the ability to design life's fundamental machines almost at will. The big question isn't really if we can gain this incredible power anymore, but how wisely we choose to use it. How do we balance the immense promise for good, for health, for the planet, with the undeniable risks that come with any technology this transformative?
And looking forward, what completely new forms of biology might we see emerge in the coming years and decades? Authored not just by the slow, meandering path of natural evolution, but by this rapidly accelerating partnership between human goals and artificial intelligence as well. It's something profound to think about.